Hi, this is Alzraa Mohammed from Egypt, and this is my fourth year in row to participate in Bioscience Initiative's virtual conference, and I'm so glad to share my work in this prestigious and so beneficial conference. Actually, as you might have seen my previous presentations in this conference, I'm interested in developing antimicrobials, and in today's presentation, I'm sharing with you my recently published article about developing the antibiotic Meropenem against the carbapenem-resistant Klebsiella pneumoniae infections. As you see here, this is my previous presentations with Bioscience Initiative conferences since 2022 until 2024. Today's presentation is titled Efficient Anti-Biofilm Potential with Reduced Nephrotoxicity of Meropenem-Zinc Oxide Nanoparticles Combination Against Carbapenem-Resistant Klebsiella Pneumonia. This work has been published in the Letters in Applied Microbiology journal in December 2024, and it is titled as a combined therapy of meropenem zinc oxide nanoparticles efficiently eliminates carbapenem-resistant Klebsiella pneumonia biofilms with reduced nephrotoxicity in vitro. In the beginning, I would like to thank my co-authors and colleagues who did their best to produce this work. We also would like to thank the Deanship of King Salman Center for Disability Research for funding this work through research group number KSRG 2024-370. And before I go through the work, I'd like to give some information about the antimicrobial resistance estimations over years according to World Health Organization and other important survey studies. In 2014, the World Health Organization estimated that the number of deaths associated with antimicrobial resistance was about 0.7 million deaths, and in 2019, the number of diseases that's associated with antimicrobial resistance was more than 6 million of deaths, and the number of diseases that was closely attributable to antimicrobial resistance was more than 1 million of deaths. And in this year, the 6 leading pathogens that were associated with antimicrobial resistance was Escherichia coli, Staphylococcus aureus, Klebsiella pneumonia, Streptococcus pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. And the number of diseases that was attributable to these 6 pathogens was close to 1 million of deaths. And according to a 2024 Lancet published study, the mortality and the number of deaths associated with antimicrobial resistance is expected to increase, and it's expected to lead to about 2 million of deaths attributable to antimicrobial resistance, and the number of deaths associated with antimicrobial resistance could reach more than 8 million of deaths. And this is why the World Health Organization in 2023 has published and prepared a research agenda for antimicrobial resistance research priorities. And this research agenda included 5 main categories. Among this category, the treatment and care category, and in the 17th research priority, there was a recommendation to investigate new antimicrobials or develop the currently used antibiotic treatments to inhibit carbapenem-resistant Enterobacterialis. And in 2024, the World Health Organization published a bacterial priority pathogens list, and this list was categorized into 3 groups, and the carbapenem-resistant Enterobacterialis was classified as one of the critical group of the bacterial priority pathogens, and therefore this was our main motivation to work on this category of bacteria. One of the well-known Enterobacterialis, Klebsiella pneumonia, and it causes several infections. First, it can cause pneumonia, which is infection of the lung. It can also cause urinary tract infection, and the infections caused by Klebsiella pneumonia is considered about 30% of urinary tract infections, bacterial urinary tract infections. And it also, in case of hypervirulent strains infection, can cause liver abscesses. And finally, Klebsiella pneumonia is also able to cause bloodstream infections. Therefore, it's so important to work on Klebsiella pneumonia infections, specifically carbapenem-resistant strains. Also, one of the main things that increases the severity and the virulence of Klebsiella pneumonia infections is their capability to form biofilms, which is an extracellular polymeric matrix that protects bacteria from antibiotics and disinfectants. And this is what makes it most difficult to treat the biofilms associated with these infections, because as we know, the polymeric matrix of the biofilm prevents the well absorption of antibiotics. Therefore, it's suggested that antibiotics could be combined with nanoparticles to enhance its antimicrobial activity or its antibiofilm activity. This is because the small size of nanoparticles, which enables the nanoparticle to migrate through the biofilm matrix and reach the bacterial cells within this biofilm. Now I'll share with you the results of this study, and as we see here, we selected three Klebsiella pneumonia strains, and this is the antibiotic susceptibility profile of these strains. And as we see here in this table, all the strains were resistant to one or more of the carbapenem antibiotics. These antibiotics, meropenem and imipenem, are the most commonly used carbapenem antibiotics. Before I go through following results and work, I'd like to share with you some facts about carbapenems and specifically meropenem. Actually, carbapenems have been approved and clinically used since 1985, and since that it has been considered as the last resort antibiotic against multidrug resistant bacteria. Years later, after the approval of carbapenems, carbapenemase producing Klebsiella pneumonia was first detected, and the prevalence of carbapenem resistant Klebsiella pneumonia have been increased specifically in the recent years and the last decade. Therefore, it's so important to work on this kind of bacteria or these kinds of infection. What motivated us to work on carbapenem and moved us to think about its nephrotoxicity is that about 70% of the intravenously administered meropenem it is excreted through the kidney after 12 hours, and this is according to the drug bank website, and this could have some negative or some side effects on patients with kidney problems, or therefore we considered it as one of the ideas that we should work on and we should investigate in this work, because as we know, on long-term use of meropenem for treating Klebsiella pneumonia or any other infection, the patients with kidney problems might have advanced medical conditions. Therefore, we considered this in our work. As we see here, we tried to make a combination of zinc oxide nanoparticles and meropenem. Therefore, we assessed the minimum inhibitory concentration for each agent alone and in combination. As we see here, we tried to test the synergism between zinc oxide nanoparticles and meropenem against Klebsiella pneumonia. And we selected, of course, the most resistant strain, which is Klebsiella pneumonia KB8. And as we see here, the MIC of meropenem when it was used alone was 600 micrograms per milli, and when it was used in combination, this number or this concentration has been reduced to 50 micrograms per milli. And of course, zinc oxide nanoparticles, when it was used alone, it was 133 micrograms per milli, and when it used in combination, it falls into 20 micrograms per milli. And according to the results of synergism test, the FICI was 0.2, which indicating the synergism between the two used agents. After that, we compared and we assessed the antibiofilm potential of each agent alone and in combination. And as we see here, the elimination of biofilm biomass matrix in the three strains in case of one strain biofilm or mixed strain biofilm, the percentage of biofilm biomass elimination has been increased in case of combination, and it reached more than 54% of elimination, and meaning that 54% of the biofilm biomass have been totally eliminated. After that, we used scanning electron microscopy to investigate the morphological changes. And as we see here, there is too much amount of polysaccharide biomatrix. And in micrograph number B, we can notice that there is a decrease in the number of cells. Also, we notice some disruption of the biofilm biomatrix, and in the biofilm in micrograph number B was treated with meropenem alone, and in micrograph number C, the biofilm was treated with zinc oxide nanoparticles alone. And as we see here, there is almost elimination of most of the biofilm matrix and some disruption of bacterial cells or damage of the bacterial membrane. And in micrograph number D, this is treated with the combination of meropenem and zinc oxide nanoparticles. And as we see here, there is almost a complete elimination of the biofilm matrix, which enabled the zinc oxide nanoparticles and meropenem to act directly on the bacterial cells and cause reduction of the number of cells and damage of bacterial membrane. Also, we tested the efficiency of this combination against a mixed strain biofilm. And in this experiment, we allowed the three strains of Klebsiella pneumonia to form a mixed biofilm together, and we treated it as we see in the micrograph number B. We treated it with the combination, and also the biofilm was formed by three strains which are resistant to meropenem and imipenem. The combination has significantly reduced the number of cells and almost completely damaged the biofilm matrix. After that, we evaluated the effect of meropenem, zinc oxide, and combination on the key biofilm genes in Klebsiella pneumonia. As we see here in this graph, it's obvious that the expression of the tested biofilm genes has significantly decreased in case of the combination compared to the other groups. And this interprets why the biofilms appeared in the scanning electron microscopy to be almost eliminated totally, and most of the cells, as we see, is damaged, as we see here. Afterward, we targeted comparing the cytotoxicity of each agent alone against virus cells, which is a kind of kidney cells of monkey. And as we see here, the combination of zinc oxide nanoparticles and meropenem has low cytotoxicity, even when the concentration has been doubled to 16-fold of the MIC. The cell viability was still over 60%, which indicating that this combination could be safely and could be more safe than the use of meropenem alone in case of patients with renal impairment. Another presentation of these results, as we see here, the red cells shows the MIC of each treatment. With the increase of the blue color, the cell viability increase, and with the decrease of the color, the color degree, the cell viability decreases. Finally, let's come to the conclusion. From this study and other studies, of course, and the World Health Organization's reports, Klebsiella pneumonia recently shows increasing resistance against carbapenem antibiotics. Therefore, this increases the severity of Klebsiella pneumonia infections, especially the infections associated with biofilms. And therefore, we recommend the use of zinc oxide nanoparticles, which, as we see in this study and other studies, of course, zinc oxide nanoparticles enhance the anti-biofilm potential of meropenem, and this explains the removal of most of the biofilm matrix and the bacterial membrane damage, as we have seen in the scanning electron microscopy micrographs. As the gene expression experiments showed, the mixture of meropenem and zinc oxide nanoparticles down regulates the biofilm genes, and this synergistic antimicrobial and antibiofilm effect of meropenem and zinc oxide nanoparticles also showed reduced cytotoxicity against kidney virus cells in vitro. Of course, this study have some limits, therefore we recommend other researchers who are interested in antimicrobials to think about or consider this combination for in vivo experiments in case of renal failure models. Also, we recommend researchers to think about developing the currently used antibiotics, maybe other antibiotics other than meropenem, and we also recommend the use of metal nanoparticles for improving the currently used antibiotics or other antimicrobial agents, since, as we see here in this work, it has an improving activity or enhancing activity that enhances the inhibitory action of these antibiotics. Thank you for watching, and see you next year.